Hi, Freya.
Hi, Matt.
Thank you so much for joining us today on Talking Academia. I wanted to ask you a bit just to start out about what your day-to-day research is like because, spoiler alert, it's quite different from everyone else who we've talked to so far because you work where?
I work at Adam Brooks in a lab, so instead of doing my research like in a library, reading different texts, reading different books, interviewing people, my research is a little more hands-on. So I go into the lab in the morning and I read a couple different papers, see what's out new in nature, what's new in science, and then I try to design a couple experiments, run the numbers, and then I get to do like a hands-on experiment to see like, oh, like what's going on in this pathway, or like, oh, if I actually add this drug to my cells, what the effect is going to be. So it's less so more of like actually trying to physically see what's happening rather than like an overviewing, like, I don't know, research.
Yeah, no, that is so cool and really different from anything I do or, like I said, most people we've talked to do. You know, you're really, like, doing your own work at the frontiers of science. I think that's really, really cool.
Thanks.
The fact that you're reading up-to-date research on a daily basis is like, I don't know, I've never lived in the moment in that kind of academic setting, so that's very exciting.
Thanks. It's very fun.
Yeah,
it's so great being able to like get up in the morning and be like, wow, someone just released a paper on like something completely outside of my field, but also like still within like biomedical research.
Totally.
And being like, wow, they found this gene that's like affected in some form of like neurological disorder. And I might not necessarily research that, but it's interesting because a lot of the times like we work with specific technologies that can actually translate to research very much so outside of what we're particularly working on. So like being able to read those papers can actually like come in handy later.
So you just know everything that's going on in science right now. No. Who knows when it's going to be relevant to your project, right?
I don't know if I know everything. In fact, like, I know a very small amount. Like, if science was like a giant cookie, I would have like a little crumb. Maybe less than a crumb.
Well, we'll see about that. I don't know if I believe it. But, so you are doing a master's, yes?
Mm-hmm.
So can you tell me a little bit about the structure of this master's? Have you been in a lab the entire time, or did that start recently, and what is coming up?
So my master's is half taught, half research. So my first term was mostly just people would come in and lecture. So we'd have 2 lectures a day, and we'd have different professors for every lecture, and they would come in and either explain something about their research, or they would explain like something that they think is very important for translational medicine. So like what we're focused on is trying to figure out how to take something that we do in an academic lab and how do we actually translate that into something clinical, because there's a big gap between doing an experiment in like a mouse model, for example, or doing something in cells and actually implementing that in humans. And like how do you figure out like how much to dose people, because like an animal is gonna respond very differently than we would, and trying to like elucidate where those differences are and how we can like overcome those challenges. So that's mostly what we talked about last term. And it was also really fun because we had a lot of like fake clinical trials that we designed. So the way the course is set up isn't like, oh, you need to like know these facts. It's more a way of thinking. So they'd be like, okay, here's like a challenge, then take the different way that people, or the ways that these professors have taught you to think, and apply that to this specific problem. So it's more like a conceptual coursework, which I really liked because I'm much more of a conceptual learner. I can't even remember what I had for breakfast, so...
That's very cool. So that's what translational medicine is? Or
So you're translating from like the lab into the clinic.
I see. Very useful stuff. So you can see kind of like how what you're learning directly impacts the world or something, yes?
Yeah, kind of. So you see more of like, I don't know, because a lot of basic research science, for example, it's like, oh, there's this pathway within the body and I'm trying to figure out like what proteins are involved, or like how they're involved, or like what structures they form. And I think a lot of the time when you're working on that, you're kind of like, well, how does this really relate to like the human beings and like the world that I live in? But I think like with translational biomedical research, you kind of form that gap. So let's say in that pathway, for example, a basic research scientist discovers like, oh, this protein's really, really important in this pathway, then like a translational biomedical researcher would go in and be like, oh, like this pathway is really important in Parkinson's disease. And be like, oh, maybe if we create a drug that targets this protein, we can actually try to like slow down the progression of Parkinson's disease or something like that. So it's kind of taking this more basic research science and how do we adapt and use that information, because it's a really powerful tool, and actually do something concrete that can affect medicine.
Very cool. That makes a lot of sense. And is it usually related to disease prevention?
I mean, it's different. My field in particular, I'm interested in gene therapy. So that's sometimes disease prevention and also like try to slowing down disease progression. So it depends on the disease and things like that. Mm-hmm. And I mean, ultimately, I think we would be able to find like cures for different diseases. But a lot of the times that's not necessarily possible with the information that we know. Mm-hmm. So just finding like some sort of treatment that can maybe like I was saying, delay progression or reduce symptoms or something like that.
Cool.
I think is exciting.
Wow, so you're doing really good work, like good person work. That's nice to hear that some of us are doing that.
You are too.
Who knows? Okay, so you had this like more taught focused part, and now you have moved into the research element of your course, yes?
Mm-hmm. So now I'm just working in the lab, so we don't have any more classes. Mm-hmm. So now we're working on like just our thesis project, which my project happens to be on pancreatic cancer, and a way to use RNA gene therapy to treat it.
Okay, very cool. I'm going to ask you to define a couple terms, if that's okay, just because myself and I hope some of the viewers don't know exactly what gene therapy is to start out. So can you give us just like gene therapy 101?
Yeah. So gene therapy it's been a field for a long time, but I'd like to say like the past 10-15 years has been such an exciting time. Like I just think it's incredible, and I think it has like a lot of potential for the future. But essentially what it is with a lot of diseases and a lot of illnesses, we can see there's actually genetic causes. So let's say, for example, in my undergrad, I worked with Huntington's disease. Mm. So that's a neurodegenerative disease. And it's been found that it's caused by this like rapid repeat expansion, which essentially just means like there's these 3 nucleotide sequences in our DNA that is just repeated over and over and over again. And then we don't have as many repeats normally as people with Huntington's disease do. Mm-hmm. So this illness is physically ingrained like in their DNA, like the basic building blocks of like who they are. And what gene therapy tries to do is we try to target that on that DNA level. Mm-hmm. So we try to like disrupt that rapid repeat expansion by essentially trying to like eliminate it from the genome. So if we eliminate it, then the idea is like, oh, if we eliminate that, we also eliminate the disease. Makes
sense. So under the assumption that certain rapid repeated expansion is kind of like a cause of the disease as opposed to like a symptom of it.
Yeah, so it's pretty much been established that like that is the cause of Huntington's disease. Okay. So another example would be like ALS. I worked on a different project with ALS. Mm-hmm. And I think it's 80% of cases or 90% of ALS cases are kind of sporadic and we don't really know why they happen. Mm-hmm. But in 10%, it's inherited. So let's say if you inherit a mutant gene from one of your parents, then you can also get ALS if they had ALS. Mm-hmm. And with this gene, we know that that is the cause of the ALS. So the project I worked on, there's this one gene called SOD1. And essentially, if you have a mutation, it's just like a 1 nucleotide mutation in the SOD1 gene. The gene codes for a protein. And this protein, if it has this mutation, becomes like a mutant protein. So it like folds incorrectly or something. And then a lot of the times, it causes these proteins to aggregate. And imagine if you're like a cell and you have all these proteins that are like clustering inside of you and you're like, I don't know what to do. So then then the cells go through apoptosis, so the cells just die. So you can actually see in the brains and in the spinal cord and in a lot of motor neurons within ALS patients and also in mouse models of ALS how a lot of the neuronal cells just die because of this accumulated protein. That's because of the mutated gene. So the idea that we're trying to do is if we change this mutated gene, then we're not going to have the protein, and then we're not going to have this neuronal death.
I see. So targeting kind of the source of the source of the source.
Yeah, exactly. So this is like the real, like the true source of what's going on. And that's the case in a lot of inherited diseases. And I think if we target that instead of just treating like later steps, we're actually getting to the cause. And that's like more progression towards actually trying to find a cure for these illnesses.
Very cool. And that makes a lot of sense. I understood what you were talking about, believe it or not. Pretty much. Okay, and you said you're working on pancreatic cancer now. So is the process kind of the same as it was with ALS? I mean, obviously different diseases, so not exactly, but...
So with my project now, it's a little bit different. So I previously worked with inherited genetic disorders. So that's where, like I was saying, you inherit the gene from your parents, and then that gene codes for like the illness or the downstream protein and everything that's going to cause the illness. But with pancreatic cancer, what we're trying to do is if you have pancreatic cancer, those cancer cells act differently than the rest of your cells. So those cells express your genes in different amounts than your other cells. So there's certain genes that have been seen to be more highly expressed in cancer cells rather than other cells. So what we're trying to do is we're trying to use like a suicide gene system. So if we introduce the suicide gene system and like link it on to these highly expressed genes, we're going to cause cellular death in the cancerous cells. But because that gene's not being expressed in the other cells, like in our regular cells, that cancer or that suicide gene isn't going to link on to anything, and we're not going to have cellular death.
Wow.
So we're essentially like trying in a specific way to just target the cancer cells to like create their own cellular death. So we're not like cutting it out or anything, but we're just like reprogramming the cells to like kill themselves essentially.
That's crazy, but really cool.
Thanks.
It makes sense. And so how do you, without maybe getting too far into specifics, get the cells to commit suicide?
So essentially we're using a system called HSV-TK. So I'll explain how that works.
I'm familiar with that system.
Essentially how it works is if this gene, TK, so tyrosine kinase, is expressed in the cell, and then we give it this drug, GCV, then I can get more specific into it, but essentially like it causes the gene to replicate DNA incorrectly. So this GCV essentially like fools the cell into thinking it's like a nucleotide during cellular replication, and like because it's fooling the cell like goes along with it, and then the cell is like, oh no, like I did something wrong, this isn't working, and then it's like, oh well, I guess I might as well kill myself. Which is like kind of a sad way to look at it, but like our cells are kind of programmed that like when things go really wrong, they know that like, oh, it's time to like let go.
That's crazy.
It came out as very grotesque. I'm sorry.
But it's for a good cause, right? We're trying to beat cancer, so like in human like forms, like more than just the cells. So it's for a good cause. It's not actual suicide. Wow. So did you choose this project, or were you assigned to it? Like, what was the process of like determining which lab you were going to be in after you finished this kind of introductory coursework stuff?
So I've been really interested in gene therapy for a while. So in undergrad, I did a lot of work, like I was saying, on inherited genetic disorders. So that was a lot of targeting on the DNA level. And I wanted to expand that knowledge a little bit more to beyond just those techniques. And I thought like, oh, 1 year master's, like this is a perfect opportunity to expand on the gene therapy field and maybe try to like integrate the two later, or try to find different ways that we're targeting these genetic disorders or like upregulation of particular genes. So this strategy is actually targeting the RNA. So DNA is translated into RNA, and then that RNA is translated into protein. Mm-hmm. So this strategy is targeting on the RNA level, which is how we're able to target with like upregulation of genes and downregulation of genes, because like all our cells have this gene, but only the cancer cells are creating them into RNA. So we're introducing this like suicide gene system onto the RNA. And the lab I work in, they're actually traditionally an HIV lab, and they have previously published on using this system for HIV. So because only like HIV proteins are expressed in cells with HIV, they're able to tag on the suicide system onto HIV-linked genes, and you're only going to have cellular death of HIV-infected cells.
I see.
So the lab, this postdoc in the lab, and the PI and I were like all kind of talking, and then we kind of came to the conclusion that like pancreatic cancer would be like a good way to move forward with this, because the postdoc in the lab was previously trying to use this for liver cancer. So now we're trying to like expand it into pancreatic cancer as well.
And how did you choose pancreatic above all else?
I mean, it's one of the most common, or becoming one of the most common cancers. I think it's the fifth most common in the UK.
Okay.
And then it's also a really severe form of cancer, so there's very little treatment for it. It's very, very aggressive. Mm-hmm. And most of the times it is fatal nowadays. Mm-hmm. So any treatment would be good for it. And I think that's why, like, ultimately that was the cancer that we decided to target.
Interesting. Very cool. Okay. And so ultimate, like, end of your master's program, you will have done what? Are you attempting to publish a paper, or are you just kind of doing your course in this lab? Like, how do you graduate from the lab is essentially my question.
So the nature of biomedical research is it takes a very long time to complete a project. So you can't really like complete a full paper in like even a year. It normally takes like 2 or 3 years to complete a project. Mm-hmm. So I'm hoping that by the end of my thesis, I'll have like the groundwork for this project. So I'll have like different ways to increase specificity. So some experiments targeting that, and different ways to increase cellular death. And then also just to show that the system works. And then hopefully in the future, they'll continue on this project and be able to implement it using better delivery systems, so using lentivirus or AAV virus to deliver it to the cells.
Cool. And where do you want to go from there? Like, are you planning on continuing this work at a different university, or do you want to be in a lab going forward? And also, if you don't know, that's okay, because I don't know what I'm going to do, so we're all kind of in the same boat.
I'm applying for MD PhD programs this June, so I don't think I'm necessarily going to continue this project, because this project kind of like belongs to the lab here, and that like this is their technology that they originally developed, and they're better set for this type of work. I think I personally am more dedicated to inherited genetic disorders, so I think I'd like to use all the information I've gained here and use this different way of thinking on the RNA level rather than the DNA level to target maybe inherited genetic disorders, because that's really important to me. Mm-hmm. And then this coming year, I think I either want to work with like policy or advocacy for people with rare genetic disorders, or for gene editing, or for people with intellectual and developmental disabilities, because that's been like a really, really big part of my life, and something that I feel really passionate for. And I think that's actually why I originally got involved in this field, is because I think it's just so unfair that a lot of people are born with these inherited genetic disorders, and like they're inherently born with this disadvantage. And like, I mean, I think our differences make us very special, and I think that's something that's really important to me. Is there's a lot of stigma around rare genetic disorders and inherited disorders, and I think that if we are able to like make everyone understand that like it's not really a disorder, it's just a difference, but like maybe we can try to create technology that can try to help as well. So both targeting from like a medical perspective, but also targeting more from like a social perspective.
Very cool. So this social perspective is maybe what you might want to focus on within the next year. So what would an advocacy or policy group look like for rare genetic disorders?
So the previous I've done is more like friendship based. So just trying to like integrate and create inclusion within the community for people with intellectual developmental disorders or just with inherited genetic disorders in general. But I also think like there's a lot of advocacy that is needed in terms of like also just like science. Like science can be very secretive, and I think because a lot of times with rare genetic disorder, like the information, there's already so little information about it that like if scientists don't really share it and don't really talk about it with each other, there's no way there's going to be like great advancement. And also, for example, like if they want a patient sample, a lot of the time, it's really tragic. A lot of times, like a family will be asked for genetic samples multiple times from different universities, rather than like now there's organizations that are trying to create like data banks that has all this information, so that universities and research groups they agree by using this information, they agree to make all their information also public. So that kind of increases just increases the ability to like move forward within the field like as a whole, rather than make it the secret competitive thing that I think sadly a lot of the times biomedical research comes over.
Right. Wow. Okay, so there are a lot of different options that you can take going forward, which is amazing. It sounds like your background kind of spans so many different equally important good fields related to biomedical research. So that's very impressive. Did you do biomedical research in your undergrad?
So I did more chem in my undergrad.
Okay.
I actually came in like maybe thinking I was going to do math. I was always really like I always really liked biomedical research, and I think that was always kind of in the back of my head, but I wasn't sure exactly what I wanted to like concretely major in. And I knew that like disability advocacy and like genetic advocacy was something that I really cared about. But I was like, I can always integrate that into like my math degree.
Right.
But then I took a math class my first semester, and I was like, I really like this. And then I took chemistry as an elective, and when I was taking this chem class, I was like, oh my gosh, this is so cool. I just like fell in love. It's funny because my mom, she's an artist, and she's like no science background. And I remember I came back for winter break after my first semester. It's like, Mom, like, look at this mechanism. Organic chemistry is beautiful. And my mom was like, this is not my child. I don't know what happened to you.
Could she not see the art and beauty in chemistry and organic chemistry?
I drew some things out, and she's like, yeah, I see it.
That's so funny. I've never heard chemistry described as a particularly like sexy or alluring topic.
Well, the thing is, like, okay, I'm very biased in this opinion, but I think like biology and chemistry are so beautiful, and like the amount of synchronization that your cells do is incredible. And like the fact that like within our genome this is integrated to make this, like, for lack of a better word, like a symphony of things that are all working together towards one cause, towards making like your cell function. And like there's proteins that are interacting like very remotely through like really complex patterns. Like let's say you activate one protein like in another cell, like in your fingertip, for example. Like if you prick your finger, that signals translates all the way into your brain, and inside of your brain it tries to think, oh, ow, that hurts. And then from your brain it translates all the way back into your hand to move it. And like if you think about that, like that's a lot of coordination, and that's just like one coordination that's happening within your body. Think about how many things inside of your entire body is like working together right now. Like think how you're multitasking, you're breathing. You're smiling, you're talking, you're thinking about your next question. You have so many things going on.
It's so true.
And like the fact that your cells are doing all of these things like in perfect coordination, that's like pretty incredible.
That's really incredible. And now I feel like I'm inspired to go out there and do the billion things that you've done.
I don't know about that, but
your 23 years of life.
But I have to say that like anyone can make a difference within this field. I think if everyone just takes like a couple extra moments to go out and like talk to someone who might be a little different from you and just like make them know that like, oh, like they're a friend and you appreciate them for exactly who they are and for their differences as well. I think everyone can make that difference. And like you've no idea how far that can go. Like just like smiling at someone or just like a really simple conversation. Mm-hmm. And I think that's like really important to me. And I think that's something we can all do.
That's wonderful. And on that note, I think we can go out and do that. So, thank you so much, Freya. This was really enlightening, and you managed to explain very complicated processes in a way that I think people will be able to understand, as well as touch on topics that are important for our day-to-day lives.
Thanks for having me.